Hello, my name is Kate Sullivan, and I'm representing the Medical Advisory Board of the Immune Deficiency Foundation. And I'm here today to talk about vaccines for SARS-CoV-2, the virus that causes COVID-19. And for the first time, I have something substantive to say about vaccines. Let's get started. So I'm just going to start with a quick reminder about how vaccines work, and I'm going to divide them into two broad categories and then come down to some specifics later on. So the first category I want to talk about, but I'm not going to come back to substantively until the end, is passive immunity. So many people that are part of the Immune Deficiency Foundation know about passive immunity. So IVIG, subcutaneous immunoglobulin, that's a type of passive immunity, giving other people's antibodies to someone to prevent infection or disease. But did you know that there are also disease-specific passive immunity products? So one would be Varizig to prevent chickenpox. One is tetanus antitoxin antibodies that are given to people with a big tetanus risk. And then for RSV for infants at high risk. So in addition to more general passive immunity protection, it can also be disease specific. And so we'll come back to that at the end. I'm going to start with talking about active immunization or vaccines. This is what many people are familiar with. So diphtheria, tetanus, pertussis, the MMR vaccine, the pneumococcal vaccine, all of those are examples of active immunization. Now, it's antibodies to spike protein that are neutralizing and protective. So I have here a very, very low-tech model of the SARS-CoV-2 virus. And these little projections that, yes, I made at home with craft clay and dowels, represent the spike protein. And I'm just showing this not to show off my artistic or craft skills, but just to make the point that we know antibodies to the spike protein are what are called neutralizing, meaning that if the antibodies, which I'm using as a clothespin here, antibodies to this prevent the virus from getting into the cell and causing infection. Now, it turns out that you can have antibodies to the tip or to the side, and there's lots of controversy about which types of antibodies are actually more protective, but antibodies to spike protein are what all of the vaccines are trying to do, because we know, at least in a test tube, that antibodies to these projections prevent infection. So that's the goal of all of the vaccines that I'm going to describe. So as I mentioned, passive immunity is antibodies from other people given to prevent disease. Now, passive immunity is short-lived. Any of you who are taking immunoglobulin replacement know that it has to be given periodically to keep the levels up. So antibodies have a half-life in the blood of about 30 days, and so any type of passive immunity is, by its nature, short-lived. I am going to come back to passive immunity at the very end because it's of particular relevance to people with immune deficiencies. So let's move on to active immunization, and the concept here is that the microbe, be it a virus or a bacteria, today we're talking about a virus, in whole or in part is administered to stimulate an immune response. Now, as I've mentioned, antibodies are the key goal of all the vaccines. And why is this? We know that antibodies can prevent infection, and we know that antibodies can mitigate the severity of infection. And it's antibodies to that spike protein that have been shown, at least in a test tube, to be the most protective. But it's important to recognize that all of the vaccines, some more than others, are likely to generate a T cell response. So for people that are antibody deficient, the fact that this T cell response can be generated is actually of relevance. So what do the T cells do? So the T cells don't prevent infection the same way that antibodies do, but they can mitigate the severity of the disease because they're already stimulated and hepped up to recognize that infection. So at the time of infection, they're already sort of primed and ready to go. So having pre-existing T cell responses can mitigate the severity. That's different than preventing the infection. So let's talk about the SARS-CoV-2 vaccines. I'm going to remind you about this website that I talked about before. So this website you can get by Googling Milken Foundation COVID-19. I've given you the URL here. It's an excellent resource. and I accessed it just this morning. So there are 197 different vaccines in development. They're not all going to make it to the finish line. And of those, 19 have entered some type of clinical trial. The remainder are just a theory in a test tube at the moment. But I would urge you to look at this website. It has really amazing resources. The graphics are outstanding, and there's a lot of great information there. I also want to introduce you to this term that you will hear in a few slides called Operation Warp Speed. The goal, it's a government program, the goal is to deliver 300 million doses of vaccine by January of 2021. Is it possible? I don't know, and I'm not going to comment on that. So there were 14 initial candidate vaccines, and the idea is that they will winnow it down to 7 liters, and those 7 liters will go into production at the same time that they're undergoing clinical trials. So in other words, those doses will be ready to go. There have been over $2 billion committed so far, and they are not even part of the way they're. Additionally, Operation Warp Speed does things that you might not think about, like manufacturing enough vials, looking at stoppers. How do these vaccines get distributed? So there's additional funding that has been allocated for the pipeline to actually get the vaccines out to people, which isn't a part we usually think about, but all credit to them for thinking about it. So let's move on and talk about different types of vaccines. The ones in red are ones where we already have existing examples, and so I would say this technology is tried and true. So I'm going to start with live attenuated. Attenuated is just a word that means weakened virus. Then talk about whole-killed virus, purified proteins, replicating viral vectors, replication defective viral vectors, mRNA and dRNA. And you may recognize that mRNA is actually the vaccine that is farthest along in its clinical trials. So let's start with live attenuated vaccines. So this is a very old method of vaccine development. And the concept is that you grow the virus in different types of cells, sometimes at different temperatures. And the idea is that you induce it to mutate into a state that's going to be less able to cause disease. So this particular method has the disadvantage of a long and unpredictable timeline because you don't know what's going to work until it suddenly shows up in your test tube. These products are usually sold as frozen or sometimes freeze-dried, so they're a little bit more cumbersome to manufacture and to deliver. These vaccines are typically not given to immune deficient patients. Some will be eligible for this type of vaccine, but not all. Examples of this are the MMR and the Rotarix vaccines. And I would say one of the advantages of live attenuated is that these tend to stimulate the best T-cell responses, which is not irrelevant in the idea that you need to mitigate disease severity. What about whole-killed vaccines? Again, this is a very old method with a lot of history behind it. Here the virus is also grown in cells, but you just grow it up. You don't have to wait for it to mutate. You collect a big pot of it and you inactivate it with different chemicals. So this is a very straightforward method, very easy to produce and distribute. Some examples of whole-killed vaccines are inactivated polio, hepatitis A, and rabies. Purified protein is another tried and true methodology. So in the modern era, this has largely supplanted the whole inactivated virus as well as the attenuated virus. So in the modern era, meaning the last 40 years or so, this has been kind of the default method. Not the only method, but the default method. So one of the reasons is it's the easiest way to produce a vaccine. And remember, you want antibodies to spike proteins, so most of these vaccines include spike protein plus or minus other proteins. So some examples of working vaccines that utilize this technology are hepatitis B and papillomavirus, or the warts vaccine. That brings us to replicating viral vectors. Now, this is the last of the tried and true methodologies. And this is a newer strategy that takes a known virus. So for dengue and Ebola, the two vaccines already licensed that utilize this strategy, they use the yellow fever vaccine. So yellow fever is a terrible disease, not really seen in the US anymore, but that vaccine has decades and decades of experience, and so they took the yellow fever vaccine extracted yellow fever specific proteins and inserted dengue and Ebola genes. And the same idea would be for SARS-CoV-2. You'd take the yellow fever vaccine virus and you would take out yellow fever genes and put in SARS-CoV-2 genes. So this is another type of live vaccine, and so probably not suitable for at least some immune deficient patients. Suitable probably for some, but not others. And this is one that has gotten warp speed funding. And then there's replication defective viral vectors. So both of the main types of this category utilize adenovirus, although two different types of adenovirus. So adenovirus is a common cold virus. And so what they've done, again, is taken out adenovirus genes and put in SARS-CoV-2 genes. And then additionally, they cripple the virus so it can't reproduce itself inside of cells. And so this makes it safer. It means it could also be given to immune deficient patient, probably doesn't induce the same level of T-cell immunity. So there's not really a history of producing virus vaccines this way, but it did receive warp speed funding. The next two vaccines utilize very different technologies than any technology that has been used before for a commercial vaccine. The first one I will talk about utilizes mRNA, and the next one I will talk about uses DNA. You will recall that DNA is what encodes our genes. Those reside in the chromosomes inside of our cell. To move that information from the chromosomes to the part of the cell that actually makes protein, there's another chemical called mRNA. And that literally serves as a shuttle to move the genetic information from inside the chromosomes out to the rest of the cell where it gets turned into protein. So this first vaccine utilizes mRNA that encodes the SARS-CoV-2 protein. So the ultimate goal is the same, to make the SARS-CoV-2 proteins that then induces immunity. But here, mRNA is the delivery system. Now, speaking of delivery systems, it's not as easy to get mRNA into the cells as it is to just inject the protein and let our immune system start working. So it's important to deliver this mRNA directly into our cells. So this particular vaccine cannot be delivered by a regular syringe, just like we're used to for most vaccines. So it uses something that looks a bit like a gun to inject the mRNA into our cells, where it will start to set up the machinery to make the protein that then induces immunity. So it's a bit more complicated in terms of chemistry. There are two main advocates for this particular technology, Moderna and Pfizer. You have probably read about the Moderna vaccine. It was recently in the news. It completed phase 2 trials where it was found to induce immunity. These vaccines have been the recipient of warp speed funding, and they are, in spite of being new technology, actually some of the front runners for large-scale rollout. This vaccine has also completed phase 1 and phase 2 trials and is also a front runner because it received warp speed funding. So that's the landscape of the active vaccines. So what about timelines? Everyone wants to know, when will it happen? And so the warp speed funding, their goal is to deliver this vaccine by January. This is a very, very, very accelerated timeline compared to what we would usually do. And I just want to make a couple of points here. So in a normal vaccine trial, you would look at 30,000 people, let's say. You would give the vaccine, you would give it to 30,000 people, you would ask, do they have side effects and are they protected? These trials are likely to be in the neighborhood of 3,000 to 5,000 people, so much smaller. So if you're looking for a side effect that happens in 1 in 5,000 people, you're just not very likely to see it compared to a much larger trial. So who will get this vaccine and when will it happen? So I don't know when it will happen. I don't have a crystal ball. Even Tony Fauci is saying that he thinks it will be completed by the end of the year, and he's much smarter than I am. So there's at least some optimism that a vaccine will be out there. So who's going to get it? So first of all, high-risk populations and first responders. No one knows how that's going to be decided, but we think that that's about 100 to 150 million people, those first two categories, and then everyone else will get it. and that's about 200 million people. So if the goal is to produce 300 million doses of vaccine by January, then that would cover the majority of the people that need the vaccine. How many people need to be immunized for herd immunity to happen? Boy, everyone's talking about herd immunity, and it's a very slippery concept right now because we do not know how effective the vaccine will be, and you cannot predict how many people need to get vaccinated to achieve herd immunity until you know the efficacy. So if the vaccine were 100% effective, you probably would need to have 50% of the population vaccinated. But no vaccine is 100% effective. So a reasonable guess is 2/3, but that's a guess. Nobody knows. And because of this concept that immunity may not be durable, it may fade over time, I think anyone's concept of achieving herd immunity is just too elusive at the moment. And remember, there's some major caveats. So while it may be possible to get the majority of the US population vaccinated, that's probably not going to happen worldwide. So yes, other countries are working on vaccines, but it's unlikely that 2/3 of the entire world will be vaccinated in a very timely fashion. Remember, some of the world doesn't vaccinate to measles yet. And so the idea that the entire world will be vaccinated to SARS-CoV-2, I think, is probably not going to be very accurate. Well, let's come down to the nuts and bolts of this talk. What about immune deficient people? So, immune deficient people probably will be excluded from any of the live viral vaccines, which is the same as it is now. Now, are there exceptions? Yes. So people with chronic granulomatous disease, maybe hyper IgE syndrome, complement deficiencies, those people can probably go ahead and get a live viral vaccine if that's the winner here, and there's no assurance that there is going to be a live viral vaccine winner that ultimately gets rolled out. In fact, there might be multiple vaccines that get rolled out. So immune deficient people can get mRNA and DNA vaccines. I've had that question before. They're not alive, even though they're genes, but they're not going to cause disease. So immune deficient patients actually could get two of the front runners right now. What about efficacy? So for people who don't have antibody deficiencies, we think that the efficacy will be roughly the same as the general population, whatever that turns out to be. The real question is for people with antibody deficiencies. So people with antibody deficiencies will likely have an incomplete response to the vaccine, which doesn't mean that they shouldn't get it. So we do think that having that T-cell response, whatever it is, would be helpful. So even people with antibody deficiencies will maybe get the vaccine if they're eligible. And I would remind you that roughly half of common variable patients respond to Pneumovax. Now, less than that respond to the influenza vaccine, so it's quite variable depending on the vaccine, but at least there's some chance that there's residual antibody production capacity in patients with common variable that could be harnessed by the body and achieve at least some antibody to SARS-CoV-2. So I think there's lots of unanswered questions, but I wouldn't say that antibody deficient patients are totally off the board in terms of getting the vaccine. Well, let's circle back to passive immunity here. So this is antibodies from other people. Many people that are part of the Immune Deficiency Foundation know all about this, but the idea is you take plasma from people previously infected and you give it to people at high risk of disease. So this is already being done for acute disease, the convalescent plasma that you've read about. But it turns out you could take this same idea and make a product like IVIG. And so that would provide some protection. Remember, it's short-lived. Right now, the convalescent plasma pipeline is really cumbersome. So this is possible, but it's quite cumbersome. Do remember that not everyone who's had COVID-19 makes high titer antibody to spike. So about 75% to 80% of people make good antibodies to spike after infection. And sadly, it's the sickest people who make the highest antibody. It does look like those antibodies wane over time, but there are companies working on taking convalescent plasma and purifying it to be delivered as a preventive product.
And I want to end with just one other concept. So once you learn the genes that are required to make that anti-spike antibody, you can clone those genes and make purified anti-spike antibody. Now this is already done for RSV virus, and so it could be done for SARS-CoV-2. And there are also companies working on that. So I think there is reason for optimism. And I'll just end with, finally, I have something very positive to say, progress on the vaccine front. And I'll just end by saying, stay strong, zebras. There is hope, and there is definitely work being done on all fronts to try and make the population safe.